Welcome everyone to another edition of the Bliss Science Cafe. As you know, we've been doing this for a couple of years now, and I'm glad that there are some people that still think that it's worth their while to log in the afternoon for a session like this. I'm really happy to have Sunil with us today. Sunil Lakshman is an assistant investigator at the Institute of Stem Cell Science and Regenerative Medicine, and he works under the InStem, as it's known, has various themes, and Sunil works with the cell fate regulation theme, regulation of cell fate theme. I beg your pardon. But what's really exciting is like very recently, Sunil was selected as one of the 9 life scientists to join EMBO, which is the European Molecular Biology Organization's Global Investigator Program. So this is really quite a prestigious selection, and so we want to congratulate you, Sunil, on that. And we're really happy it's come at such a great time, and really happy that you could be part of the Science Cafe with us at this great time. So everyone, if you want to congratulate Sunil, please go right ahead. I also want everyone to know that the Science Cafe is not your average science talk. I mean, it is in some way, but the way it's different is we encourage disruptiveness and we encourage an extremely informal atmosphere. So please do not stand on a formality. If you have a question to ask, please feel free to interrupt Sunil and say, hey, what did you mean by that? Please treat this like your own little living room. Get yourself something to drink and sit down and just enjoy the afternoon and interact with the others that are here as well. And having said that, I will pass it on to Sunil now. Thanks so much for being with us today, Sunil.
Yeah, thank you, Maheen. Thanks, everyone, for being here. I'm just going to start my screen share. Let's see if it comes. Okay. Um, Hopefully, you guys can see the screen.
Yes.
Yeah, good. Okay, so let's get started. And thank you so much, Maheen, and the communications team, Pavitra, Amrita, Chandrakant, for inviting me to do this. So, thank you everyone for joining. And like Maheen said, this can be more of an informal discussion, which means feel free to shoot any kinds of questions you might have or that arouses your curiosity related to the topics we are talking about today. And we could have this in a very conversational mode. So, what I thought I'd talk about is something that's fascinated me for a long time. This is not what you'd say the core research of the lab, but this is something we peripherally dabble with, which is to understand life in very extreme conditions. Now we know that um, uh, most cells, most animals, most organisms, most plants like to live in what you'd call nice conditions, pleasant weather, lots of water, lots of food, and so on, right? But we also know that life exists in places that you think life should not exist. So there must be some unique biology, some unique chemistry that allows these organisms to work. And so, what we'll do today in this Science Cafe is kind of dig into what those little processes are inside the cell that enable life to exist in very extreme environments. So, before we get into that, we actually need to have a good definition for what is life. And although we all think we know what's living and what's not, um, there's actually no real consensus on what is life. Uh, if you look at life, it's some property that distinguishes the living from the dead, and whatever is dead is being deprived of life. So you're making a nice little circular argument on what makes us and the average amoeba the same or different, right? So this is James Gould's famous explanation of life. But largely, we can agree that for life, for anything to be considered alive, it has to have the capacity to carry out three what you'd call basic activities. One is metabolism, one is self repair, which is dealing with the ability to fix itself, and this very important thing called replication, which almost everyone knows about because of DNA and the genetic code of life. So essentially, what you have though to declare something to be alive is something like this little circle over here. If you're alive, you have some ability to do metabolism, which is make, break energy, grow, and so on. You have some kind of cellular form and structure because the cell is the simplest unit of life. You respond to changes in the environment one way or the other, either overtly or covertly. You have growth, you have development, you have the ability to reproduce, at least for some part of your life. And when you reproduce, there is heredity, which means your DNA, your genetic material is passed on, and you have homeostasis. Homeostasis is to have some kind of a stable internal condition that you manage to maintain. This is what you'd call what life is, right? And if you think of life, the little unit of life is the cell. So for anything to be alive, the cells inside it has to be alive if it's a multicellular organism, which is us and mammals and plants and insects and worms and so on. Now the cell is an amazing entity. It's not just a little bag, but it's this gorgeous illustration over here by David Goodsell. Tells you how complex a machine the cell really is. It has a cell, some organisms have a cell wall, all organisms have this complex cell membrane. It's filled to the brim with all kinds of proteins as well as little molecules inside it, all of which are not just sitting there static, but they're dynamic. They're moving around and doing something all the time. If you want to see how dynamic the cell is, I would encourage you to see this movie called The Inner Life of a Cell. It's very much in the Attenborough style of natural movies, like The Wild Planet or something like that, but it's inside the cell, and it's probably the most accurate description of a cell we have out there in a video form. So this is on YouTube. It's called The Inner Life of a Cell. And one very important aspect of the cell is this cell membrane over here, which is made of these kind of molecules. They're called phospholipids. They're charged, partly charged and partly entirely uncharged, and form a little layer that keeps it like a nice active bag. So it's not a static plastic bag, but a porous bag that allows some things in and some things keeps them out, and keeps all these gorgeous proteins inside the cell. Now, for a cell to be alive, this entity is quite fragile. It's quite delicate. In fact, one of the easiest things to kill in a lab is a cell. You put anything on a cell, salt, too much water, little bit of acid, just a little bit of acid, remove some glucose, the cell will die. So this is a fragile little entity. But this is what makes a living creature, right? And what makes a cell tick is this word that you would have all heard about. It's called metabolism. When we think of metabolism, we just think of something: slow metabolism, fast metabolism, metabolism that doesn't give you enough energy, feeling weak, feeling tired. That's not what metabolism is. Metabolism is the chemical basis of life. This is really all the reactions in a body, in a cell, that are carried out. So you have both breaking of molecules and making of molecules. You make food to energy. Energy has to be a usable chemical form, and that's what happens inside cells. You make building blocks for a cell, which would be things like proteins or lipid, fat, or sugars or carbohydrates. All of these build the blocks of a cell. And you have enzymes that carry out this reaction. Enzymes are special proteins that allow molecules to be made or broken. And this is what allows cells to grow and reproduce and so on. So really in metabolism, you have just two simple parts: anabolism, where you make bigger molecules from smaller molecules. So for example, you have amino acids, they form a chain and they'll form a protein. It's anabolism. And catabolism is large molecules being broken down into small molecules. So you have large carbohydrates, you'll break them down into glucose. Glucose will give you energy. So that's the whole process. And you're making energy, you're using that energy to make these large molecules, and then you're growing. So this is metabolism. Now, in all of this, one of the most important things for life of a cell is water. And we all know water is an unquestionable entity for life, right? So why is water so essential for metabolism and therefore life? We've defined what life is. You need metabolism. And for metabolism and also for the integrity of a cell, you need water. And water is a very special molecule, and which is why Earth is a special place, because we have lots of water. So it's ideal for life. Water is actually a very simple molecule. Everybody knows H2O is water, but it's also got some very unusually special chemical properties. One of the few molecules that does so. It's a polar molecule. There are many polar molecules, but this is a nice polar molecule in that it's neutral charge, but it has a partial negative charge on the oxygen and a partial positive charge on the hydrogens. This makes it good for many things. One thing makes it very good for is it's a great solvent, which means many things, many minerals, many ions can dissolve in water, which means you can move things around within the cell or within a tissue. It also has this property of hydrogen bonding very easily, so it makes it cohesive. It makes it stick to many things. It has a high heat capacity, which means it doesn't heat up or cool down very quickly. This is very important because you need to maintain an ambient temperature. I mean, the reason lakes or rivers largely remain at the same temperature is because of this heat capacity. It's the same with this high heat capacity also has a high heat of vaporization, which means when it evaporates, you have cooling associated with it. This is important for life on the planet. And it has this other special property. It is less dense when it becomes a solid, which is ice. And you know, your 7th standard physics textbook tells you something about anomalous expansion of water. But this is important because now when water freezes, the top layer will freeze first, and it will protect the rest of the water from freezing. So this makes water very special. And water supports cellular structures, right? Uh, which means if you have a bag and it's filled with water, it'll remain turgid and nice and round. You remove water, it'll deflate or go away. There's another thing because of its special chemical properties is that it's central for metabolism. This is through processes called hydrolysis reactions. So, for example, if you look at this horrible chemical structure, this is actually sugar, sucrose. And this is how you break it down to get glucose. And for that, you need a little bit of water. The water adds this hydrogen over here, you get these hydroxyls, and you make this molecule sugar into glucose, which gives you energy. Or you have other reactions in the body like this. This is an aldehyde. Aldehydes are nasty reactive molecules. They have to be gotten rid of quickly because they'll cause damage in the cell. With water, you hydrolyze it to an acid, which is much easily dealt with within the cell. So water is the special molecule. So now we have talked about life in general. You need to replicate and do all this metabolism. And for metabolism, you need water. So you can't have life without water. But what we're going to talk about is the amazing property of many, many cells and many, many organisms to live essentially without water for very, very long periods of time. And how is this possible? There are places, even on Earth, where there is water, but it's not in a usable form. So you have steam vents where the temperature is 100 degrees, it's just steam everywhere. Or you have deserts where there's very, very little water. And even when it comes at very small periods of time. Or you have ice in the Arctic or in the Antarctic or on top of very tall mountains, but where the water is frozen, so it can't participate in this nice chemistry. But we know this very well that in all these places, they're teeming with life. So this little steam vent you see, this is in Yellowstone, and all this orange and blue and yellow that you see are actually not because of the soil, but because of microbes that grow in it. These are extremophiles. These are little bacteria or archaea or occasionally fungi and algae that grow in the steam vent at 100 degrees. If you lift a rock anywhere in the desert, you'll find it covered with these lichens, fungi, algae sometimes, cyanobacteria. They're all there growing on the rock in the desert. So how do they manage life without water? And the reason this is interesting or exciting is that if we can crack this, we figure out the rules of extreme life, which then makes us think about the whole universe as a place where you could potentially live in. So eventually we're going to end up talking about life on Mars. And how is that possible?
Sunil, can I jump in and ask a
Yeah, please
question
do.
here?
Please do.
Yeah. So Atharva asks, is there any other compound which can or has replaced water in any organism?
Aha. No. The short answer is no. Because a hydrolysis reaction by definition requires water and there is no metabolism without hydrolysis. But what we will talk about are molecules that help a cell survive in the absence of water till water comes back. So you can live for long periods of time in a way, you're still alive, suspended animation without water. Okay. But there is no molecule, because of its chemistry, there's no molecule that can replace water. So which is why, uh, as far as we know it, the way we can define life, you need water. And in the current definitions of life, you need carbon-based life forms because of the way we think of energy. And we need oxygen, at least today, for life. But you can have life with very little or no free oxygen. This is still possible. Okay. So nothing has replaced water, and nothing can replace water. The others are negotiable. You may be able to replace carbon with something else. In fact, there's a famous Star Trek episode where they replaced carbon with silicon in some planet. And you, in theory, can have life without oxygen, but you can't have it without water, at least the way we know it. Okay, so what are the challenges for life without water? Life as we know it, which is the life of a cell. This is this one point I pointed about earlier. If you have water, you have a nice little swollen balloon of a cell. If you remove water, the cell will shrink and shrink and shrink. The pressure outside is far greater than the inside, and the cell will crumple and destroy itself. The other piece that's essential for that requires water is hydrolysis, right? And these reactions that require water. So metabolism, to make energy, to make molecules, to make proteins, you need water. So how can you have life without usable water? That's the question. And we know there is life without it. Well, there's a bit of a trick over here, because life that we know without water is essentially a state where all the definitions of life barely hold together. So the definitions of life are these, right? You have to do all these things: respond, do metabolism, homeostasis, reproduce, divide. Well, when we don't have water, you have organisms that go into the state of suspension, uh, uh, almost to the point of them practically not being alive, because at that point of time, they don't do any of this. But the amazing thing is they can come right back and do everything else rather quickly. So, yeah. So that's what we're going to now talk about. And then from there, we'll try to think about how knowledge or understanding of this will be useful for us. So coming back to life without water. Without water, since we can't do metabolism, what organisms and cells have to do is shut down metabolism. So which means if they have to deal with life without water, they have to find a way to shut down everything, but in a way that allows them to come back to full functioning when water comes back into the system. So you shut down all of this metabolism by two kind of methods. Essentially, what you do is divert all energy making processes to storage processes. So what you do typically, for example, is if you have food, carbon, glucose, sugar, something, you'll do these chemical reactions like glycolysis or respiration. Everyone would have heard of respiration. You make energy. ATP is the currency of energy. The moment you start reducing water, the cells that manage to survive are able to divert all this action away even before all the water disappears. So there are ways by which cells sense this and transfer it to what you'd call storage, put things away for a later day. We're going to get into how they do this. Essentially, what cells do are two things. And this comes back to the previous slide also. You have to keep the cell intact, right? You can't allow it to burst, because once it's burst or once it's broken, that's it, it's dead. There's no coming back from a completely destroyed cell. So you have to keep the cell intact. So essentially, everything they do is to divert resources to make molecules that might be what you'd call storage molecules, but allow cells to survive and prevent them from exploding or crumpling. So they do two things. The first thing they do is convert things like glucose into what you'd call non-reducing complex sugars. These are two very famous molecules. One of this is common sugar, the standard sugar that you have in your tea and coffee, right? Sucrose. The other one is a very closely related molecule, which is trehalose. In fact, this is the popular way by which many cells survive desiccation. We'll talk about what these sugars do, but by and large, what these sugars do is replace water within the cell at very high concentrations. Some of you might have seen this if you're amateur cooks. If you make a sugar syrup, as you make it thicker and thicker and thicker, at some point, when it's thick enough, it forms a little gel, a little sticky gel that's not quite solid, but it's not quite liquid either. That point, it has very little water in it, but it has phase transition. It has gone from being in a liquid form or a crystalline form to this gel-like form. And that's what cells do. They make so much sugar or so much trehalose that in some ways, inside the cell, you become a bit of a gel. And that can protect the membrane from breaking up because it's this little gooey, syrupy, stringy thing that can protect the cell. The other thing cells do is make special proteins. Now, of course, we know the cells are full of proteins, and these proteins do different things inside the cell, right? They do metabolism, they do information transfer, they allow DNA to replicate, and so on. And all proteins have structure. Structure, uh, uh, I just had some questions. I'm sorry, I just got distracted by some questions. I'm going to read out the questions before we race ahead. So, one of the questions is a tree exchanges energy with the environment, but a wooden table does not. Does this point toward fluidity, need for water or liquid as an irreplaceable requirement for life? Um, yes, but hold the thought for a little bit because we'll expand this definition of living and dead. Okay, so we'll come back to this question in a bit. Another question, which is a really nice one: if cells can survive for long periods without water, did early life forms evolve without water? Okay, this is a loaded question, but as far as we can imagine, we think all early life might have uh, evolved with water. In fact, they must have originated in what they imagine to be deep aquatic vents or places like that, where you had a lot of minerals, but you had a lot of water. So that's the idea. Of course, um, the origin of life is a widely debated field, but what you're trying to imagine there is possible chemistry. And in all these explanations of origin of life, no one's been able to imagine it without water. So speculatively, answer is no. Another question, nice question, is water a requirement for phase transitions during energy exchange metabolic processes, which cannot occur without water? Actually, it's again an interesting question. What happens when you phase transition proteins is you squeeze out water. Um, is it an absolute requirement? I don't know actually. So let's see if this makes sense at a later part of the talk. We'll come back to that. Okay, good.
Sunil, one
Yeah.
second. Someone on YouTube is asking us, you mentioned pleasant and extreme conditions, and that might be opposite for different organisms. So, isn't extreme or pleasant a subjective matter for different organisms?
As far as so I'll make this a very uh, um, unbiased statement. As far as chemistry goes or biochemistry goes, there is actually a fine range of what you'd call pleasant. So most proteins, most enzymes work very well at some temperature between 20 and 40 degrees, which is why most warm-blooded animals maintain a homeostatic temperature of 35, 37 degrees, right? This is what we are all at. There's good reason for that. The kind of chemistry that is required for metabolism is ideally functional in that temperature range. Now, at very cold temperatures, most of these proteins cannot function at all. Essentially, they're static. So you can't have life. It's really unpleasant, impossible to have active life at those temperatures, but there are some exceptions. The same goes for very, very high temperatures. Now, there are organisms that survive in these very high temperatures, and they have made an evolutionary decision, kind of. I mean, it has happened because life is possible there, but there are real challenges to carrying out biochemistry at very high temperatures because a lot of chemical reactions that we think of as essential for making energy and so on are very hard to do at those temperatures. Okay, so because we're kind of skimming through the surface, that's the nutshell answer. There is a broad temperature range which is ideal for life, which is in this 15 to 40 degrees range. At colder temperatures, organisms have to maintain a cellular or body temperature that's higher to enable this good biochemistry. And at hotter temperatures, they have to find ways to uh, combination deal with what things like denaturing proteins. At high temperatures, proteins unfold. They have to find ways to protect that and also reduce their own internal temperature. So maintaining body temperature is one of those amazing things that happened along evolution, and that's what allowed this huge diversity of life, body or internal temperature. Plants can also maintain it; they don't have bodies the way we think about it. Okay, so let's go on. So we talked about sugars that help you survive. These are common sugars, your popular sucrose sugar and trehalose. The other thing that some cells and organisms do are make these unusual proteins called intrinsically disordered proteins. It's a fancy way of saying proteins that are shapeless, that are wobbly. Normally, proteins are nice. They have a nice shape, a nice structure. So some of you might have seen it in high school or in college, the structure of hemoglobin or myoglobin. It's a nice, compact, ball-like thing. There are many proteins that actually don't have good structure. They have these floppy little globes all over the place that are wobbling around. Now, on their own, in small amounts, they don't do much. But if the cell makes a lot of them, these proteins come together and they kind of glue up and again form a little gum or a gel or a little paste or grease. You could think of it as grease. You have a lot of fat or a lot of grease. You all know that it's very hard to kind of freeze things and so on and so forth if you have a lot of gooey grease around. And that's what they do. So these proteins can actually help protect the other proteins in the cell that are required or protect the DNA or protect your RNA. So that's the strategies by which cells do this. But that's kind of vague. What I've said is kind of vague. Can we learn a little more about specifics and how we do this? We do. And we are now systematically figuring out what each of these molecules do. Uh, Now, the previous things I talked about, sugars and protein, are especially useful when you lose water. But the same rules seem to apply when you have water that's not usable. So this is this point of frozen conditions or extreme heat. Some organisms, animals, for example, insects and so on that survive frozen conditions, also make different types of sugars. In this case, there's a minopyrosyl xylopyranose. It's another type of a sugar, seems to do something rather similar to sucrose and trehalose. And they also make different kind of low complexity antifreeze proteins. These proteins actually prevent water from crystallizing by holding on to lots of molecules of water and separating them out. We talked about how water comes together with hydrogen bonds and so on. Antifreeze proteins prevent water from doing that very well. So this little beetle, for example, can survive rather well through an entirely frigid winter in the tundra. So that's great, right? Now, I'm going to shift a little bit now to little details of surviving and how we figure out these little processes that allow you to survive without water. So there's a little take home right now, just a little summary. Not all cells can survive without water, but there are many cells, many animals, many organisms that actually do so rather well. And the way they do that seems to be by using these sugars as well as these proteins. And how did we figure all of these out? A lot of this figuring out has happened over the last, let's say, 20 to 30 years. 30 years ago, it was purely descriptive. But thanks to advances in our technologies, now we do genomics, we do proteomics, we do high throughput genetics. These are all fancy words of saying we can break apart a cell and put it together again with a fairly high degree of precision or understanding. And to do this, we use models or cells or organisms that are easy to manipulate in the lab. Now, my own favorite model is yeast because we use this in the lab, but that's not the reason it's my favorite. It's my favorite because it's just gorgeous. This is a sculpture of a yeast cell. It is what you'd call a prototypical eukaryotic cell, which means we learn a lot about how all cells work in eukaryotes using yeast. Of course, most of you are more familiar with using yeast as dry yeast for baking or in the form of beverages. Uh, uh, and right there, you see that active dry yeast itself tells you that yeast can survive for a very, very long time in a packet. There's no water there, and these are completely dried out. Now, the beauty of yeast, though, is you can do all kinds of genetics with it. You can put it in all kinds of conditions. You can grow it really easily. Everybody can grow yeast. And you can systematically test out what molecules or what processes are involved to survive desiccation or the loss of water. And you can also use it to study other water related processes. Freezing, what happens when you freeze it and thaw it out, freeze it and thaw it out, freeze it and thaw it out. What happens when you put it in very high temperatures? Can you put it straight away or do you have to condition them? And using this, we could work out many of the rules you need to study life where you lose water. And a lot of common themes came out from this that amazingly have held up in almost every organism that we have now studied. The first thing is, for most cells or most organisms to survive in the absence of water, they all shift to a low metabolic state. This is universal now. And we started learning these from yeast, but now the same rules seem to be functioning in all kinds of organisms. We talked about sugar, the sugars and trehalose in particular, acting as a protectant. What we learned from yeast is that it works beautifully as a membrane protectant and protein protectant. And we found that cells also do something called lipid metabolism, that is, changing the composition of fats within the cell. And this is important because lipid metabolism is very important in protecting the membrane of the cell. As we studied more and more processes of life without water, we found that there are these biochemical pathways that for a long time textbooks have said were useless. It is unclear why these pathways exist. These pathways are extremely important for surviving stress where water is lost. So something called the glyoxylate shunt, for example. These pathways are not essential for normal life. What they're essential for is to divert resources to make protective molecules under stress. The other thing we learned is that cells can survive a very, 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 very long time in a desiccated form, which is basically suspended animation. This desiccated form without water has no signs of life, but comes right back when you put back water. So all of this, the beauty of all of this is you can use a simple model organism, something like this Golgi yeast, and carry out experiments in the lab and systematically dissect out processes. Okay, so let's see if there's another set of questions. Aha. Somebody has asked, would the IDP transition work? These intrinsically disordered proteins work if they transition from a complex 3D structure to a simpler globular structure, assuming they might not really function that much. So, without sticking my neck out too much, this is a hotly contested and debated field right now. Answer is unclear. A lot of people would say no, but some people are saying yes. So debate the jury is out there, and I think we'll uh, know why this happens. Okay. Pratik wants to know, is the presence of trehalose in the hemolymph of cockroaches a major reason for them being so cosmopolitan? Okay. So cockroaches have a bunch of complex sugars. They make a little bit of trehalose, but they don't make that much as many beetles, for example. And chytrid beetles are very good at surviving freezing temperatures and so on. But cockroaches have a far more complex sugar metabolism than we would give them credit for. So they make what are called complex glycans and glycoproteins, which are proteins conjugated with sugars. And it looks like those are rather important in their ability to survive all kinds of conditions. But yes, cockroaches are indestructible, although they are not true extremophiles in that they cannot live in conditions where there's absolutely no water, in absolutely very, very, very high heat or very, very arid conditions, or deserts or frozen deserts. Okay. But the world champions of surviving extremes are what are called microbial spores, especially bacteria, but not just bacteria. Similar processes happen in other microbial cells, some fungi, archaea, and so on. Slight differences, but essentially what these bugs do, and there are now millions and millions of them, thousands of different species, is that when they are stressed, where food is removed primarily, they go into a complete ametabolic state, shut down metabolism completely. And what they do is divide off all the important parts of the cell and compartmentalize it. So this is a cell, a bacterial cell, normal cell, dividing, happily growing, blooming everywhere. But when you take away all their food, they compartmentalize all their DNA into a little pocket, which is surrounded and filled with this molecule called dipiclonic colonic acid. It's full of that, and that seems to protect the DNA. And the spore comes out. Everything else goes away. Now the spore is truly suspended animation because there is absolutely no metabolism in them. and they can live for a very, very, very long time. They're rotationally immobilized. That means there's no motion within them possible. They've excluded almost all the water. So this is almost a hard, compact little entity. And water can barely get in. That might be because of the composition of the outer membrane and the inner membrane of bacteria. And the lipids or the fats are also immobilized. So essentially what they've done is compress themselves into a little indestructible bomb shelter bunker to survive any stress, not just the loss of water, but they also survive freezing and loss of water and so on forever. Now, this is the amazing part. How long does one think spores can survive? For a long time. We thought, okay, spores can survive a few months, maybe a few years, and so on and so forth. But there's this really interesting scientist whose name is Raul Kano. He calls himself a paleo microbiologist. He's an emeritus professor at California Poly. And essentially, what he did was what they wanted to do in Jurassic Park. He isolated bacterial spores from amber found in the Dominican Republic, which was dated to at least 25 million years. And he found these spores inside, and then he revived them and they grew. So essentially, you have Jurassic era bacteria that can be revived. These are not bacteria that have been growing and dividing since then, but were trapped in amber 25 to 50 million years ago, and they survived. And what he did was exactly what they wanted to do in Jurassic Park. They found amber with a bee or a beetle. I think this was it. This was a bee in the Dominican Republic trapped in the amber. It was completely fossilized. Found the amber, kept it very, very, very sterile, cracked it open, dug inside the bee. So it's not even the bacteria on the surface, but it's bacteria isolated from inside the gut of the fossilized amber bee. Put them in growth medium, which is your standard bacterial culture medium. And these spores germinated and became bacteria again, and they're growing, as far as I know, till date in his lab, revived them. So really, we have in some ways shifted the definition of life completely. You can stay in suspended animation for 25 million years if you are a bacteria. And it's not just bacteria. He did something even cooler. he, Raul Keno isolated yeast from amber from some bees, which were again trapped from 25, 30 million years ago. And he revived these yeast. These are Saccharomyces yeast, just the kind we use for baking and brewing. But they behaved a little differently. Of course, they haven't been evolving they missed out 25 million years of evolution. So they behaved a bit like ale yeast, a bit like lager yeast. Does not go dormant, but he managed to make beer out of them. And you can buy fossil fuel beer in California if you live in Central California. So great. So actually, you might even find something useful from reviving Jurassic era microbes. But that really tells us how far we can go with surviving extreme water loss. But the point here is that most of you must have caught on already. Bacteria, yeast, these are all just single cells. They're not very complex. But if you want a whole animal to survive, or an insect or a plant, it has to be much, much more complex, or you would imagine it. We always attribute greater functions to nervous systems or greater functions to complexity. Are the rules different now for surviving extreme water loss in an animal or in a plant? Well, turns out it's rather interesting. Of course, we know that there are a few plants that can survive extreme water loss. This is something called the Jericho rose. It's a desert plant found across the Americas. It can survive as a crumpled, dried up little ball for years, and you add water, it comes right back. But then we have these world champion animals of surviving water loss. So you have the toughest animal on earth, these cute little things called water bears or tardigrades. They look quite impressive here, but they're actually not that impressive. They're less than half a millimeter in size, and they look like this only under a microscope. They're cute, but not that impressive otherwise. You have rotifers. which are again fairly simple animals, but they have a brain, a digestive system, a circulatory system, and so on. You have shrimp. They now survive extended desiccation without water. They can, in their desiccated state, survive anything. So tardigrades have gone to space and come right back, and they're just fine. And how do they do this? It turns out that the rules for them surviving are quite similar to the rules that little bacteria and microbes use. So I'm going to show you a little video to show you how we revive worms in the lab. And this is work done by my collaborator and me. So this is actually kind of work that we've done. I'm going to now share something else. I am going to share a little YouTube video of how we resurrect worms from the dead. And these have been worms that have been um, sitting after drying out for many, many months. So just watch this video. There's your dried up worm. You add water and you wait. And 20 minutes later, the worms are wriggling. And after a couple of hours, they're all swimming and ready to get out of the water. And very, very soon, a few days later, they're back to be reproducing and growing and so on and so forth. Quite happy. So the first time we saw that, we were quite amazed by it. We had no idea that they would do this. But they did this rather beautifully after 6 months sitting in the freezer. So I'm going to go back now to my presentation. Great. Yeah. So turns out that organisms which have a brain, a digestive system, a nerve system, thousands of cells can also survive desiccation rather well. And what was more amazing as we started to systematically dissect out what's going on is that the rules of this seem to be the same as what you'd call a rather uninteresting or simple microbe. Essentially, the cells in this organism coordinatedly do the same thing. What they do is shut down metabolism. They make lots of these intrinsically disordered proteins. They make sugars. In this case, the worm makes trehalose. They detoxify certain things and they shut down a lot of oxidation. But there's a little twist here. No animal can survive instant desiccation. What they need to do is be preconditioned. They need to slowly encounter conditions where water is slowly being removed. If you very rapidly remove water, they'll all die and they can't come back. And that tells us that the processes for a multicellular or a complex organism are much, much more regulated and have to be coordinated for a long time. And the cells, some external neuron or something like that has to sense this change in the overall environment, start processes to make all these molecules. And once done, they can then survive desiccation. Now, of course, this makes sense when you think about it. No plant or no worm is suddenly going to go from being in environments where there is water to no water. They will first expose themselves to conditions where they see the water loss and then develop ways to do it. So collectively now, what the worm and tardigrades and plants teach us are there are two parallel processes. You have to protect the membrane. To do that, one of the best ways to do it is to use a sugar. The reason the sugars work really well is that they displace water and take the place of water while also at high concentrations becoming a bit of a gel. The intrinsically disordered proteins end up at high concentrations to do something similar, phase transition, but they also create protected spaces for the other proteins in a cell. Okay, great. So now we have fossilized beer now, right? From yeast from 25 million years ago. How long did we think a worm can survive? In our lab, it was 6 months or 9 months or so right now. We now have rumors that somebody has preserved worm for 2 years. Great. But what uh, uh, blew our minds recently is a report from Russia from last year or 2 years ago. Now, these look like the worms that I just showed you, except that these were isolated from the Siberian permafrost. They did it about 6 years ago, and then they translated the Russian paper into English, and suddenly all of us realized that you could dig deep into the permafrost. This was 30 or 60 feet into the permafrost, and this has never been thawed out. They isolated worms from there. They put these worms in a nice sucrose solution along with a few other goodies and food. And a few days later, they were back and swimming around. So now we have worms from the Pleistocene, which we can compare to worms of today. And first of all, we can find out how they're different or the same. But we now know that suspended animation in conditions where there's no usable water or where there's no water is actually possible for complex multicellular organisms, not just worms. So, really, what we have now is that we can imagine life potentially existing for thousands and thousands of years in a suspended form. And this brings us to the last part of the talk. Now, we talked about extreme survival without water, without food. That's what you'd call cryptobiosis, essentially, raising the dead. There is no metabolism, no signs of life, and you can still come back to life. But now we have a much, much more wider common phenomenon. It's called a form of dormancy. And this has a spectrum in biology. You have hibernation, which many of us have heard of. You have torpor, which is very, very common in animals, as well as diapause, which is very common in developing embryos. Now, all of these are not as extreme as what we talked about, complete loss of water and so on and so forth. But in these conditions, you have many features that are actually similar to what happens over here, just not as extreme. So hibernation and torpor in particular make us now start thinking about suspended animation for a long, long periods of time. Now we know hibernation is very common. Lots of rodents, squirrels, chipmunks, groundhogs, they all do this across winters. Larger animals, bats, bears, are all able to hibernate. Now, you and I are not able to hibernate, and a lot of other animals are not able to hibernate. What happens during hibernation? Hibernation seems to be this intermediate between just a cellular process and a whole physiological process where you manage both aspects of it. So, now in hibernation, a key feature of hibernation or torpor, this whole spectrum, is that your temperature, the homeostatic regulated temperature, drops from 35 or 37 down to around 10 degrees centigrade. So that means you slow down your metabolism a lot. You're not ametabolic, but you're very, very slow metabolic. And with hibernation or with torpor, you have periods of activation over a long winter, let's say. So this is October to March or April. And you see that every month or so, there's a periodic spike in temperature and activity. And this is what mammals use to kind of replenish themselves during that long winter sleep. So they're in deep hibernation. They kind of come out of it, go back, deep hibernation, go back, come out of it. And that's how they survive. And thanks to modern methods now, we can understand the whole physiology of what's going on. So we can look at metabolite changes, metabolism changes, and how proteins change. And what we're starting to see now is quite beautiful. So for example, this is the cycle of proteins that are up or down when you're active or inactive. And you can see it beautifully changes over this whole period of when you're active, when you're inactive, when you're active again, when you're inactive. And you come up and down and up and down. We're starting to get a grip on hibernation and torpor. We're starting to see now that some things are the same as what you see in extreme survival, but some things are a little different. First of all, you don't typically see accumulation of sugars like trehalose and so on. They don't seem to be there. But you do see changes in some proteins. It's still unclear what they are. There are chaperones that change. We don't see as much changes in intrinsically disordered proteins and so on and so forth. But still, there seem to be ways by which um, um, some common ways by which cryptobiology and hibernation have some things in common and some things are different. So now can we work out the rules that are common and some that are different, right? So essentially now we'll summarize this part of the talk. So to survive suspended animation for a long time, you go into a very low metabolic state, make very large amounts of very few molecules, these intrinsically dissolved proteins, these sugars, and make molecules that protect the cell. But the idea of dormancy and hibernation and torpor are kind of an intermediate stage. It's neither here nor there. You're neither in heaven nor on earth. You're right in the middle, and you can survive very long periods of time. There's actually uses now for understanding all of this. So like we saw, we can make fossilized beer, but I think more than that, we have ways now to think about preserving organs after accidents. Let's say you want to organ transplant. You can start now thinking of the rules of how you keep cells and organs intact, say by using these intrinsically disordered proteins or sugars to keep organs intact, which could be used for transplantation later or preserved for future use. So these are very practical uses. We could preserve plants, protect plants and seed lines through drought. These are things that we could think about. And of course, there's the most exciting part, which is to go to Mars, right? So all of you who've seen Star Trek and Star Wars and whatever else know that you have cryo chambers where you just go to sleep and then wake right up. Might not be as absurd as it used to sound. Uh, and there are real efforts now to create torpor-like or hibernation-like conditions for potentially creating cryo chambers, with which you will eventually go to Mars and infinity and beyond. And so uh, so now we can have, I think, open questions. Maybe the moderators can read out some of these
Yeah,
questions. That
I
would be
think
better.
coming to the last thing you mentioned,
Yeah.
cryo sleep and all of that for humans. So, Sriram asks, how do cells and organisms like humans that lack molecules like Trehalose deal with fluctuating water levels?
It's a good question, right? So you might have cleverly noticed a common theme across over here that the organisms that survive extreme water loss are not very good at moving around. So most microbes are where they are. Plants are rooted to a spot by definition. Worms and nematodes can't really go very far. So if the pond dries up, they're stuck without water. And that seems to be a fairly common theme that mammals and birds that can move around have lost this ability to survive water loss. That's mostly because they get up and move to places where there's water. And that's been the basis of a lot of life, birds, mammals, and so on. Now, the interesting question is, why was there evolutionary selection to lose it? And is it possible to regain it? Now, regaining some of this may not be as trivial as it sounds, but at least at the level of cells. So, for example, platelets, uh, people have engineered platelets to make trialos. And once they make trialodes, platelets can be frozen and kept for a very, very long time. So it can be useful medically. But will we regain it? Now, probably not. You can engineer, I don't know, sometime in the future, you might have engineered animals or humans with making trialos and making IDPs and surviving desiccation. But otherwise, we don't deal with loss of water very well. And this is what we think of normally in life. Even most plants don't deal with complete loss of water very well. They die.
Okay. So coming to another question about, what is life alive and what's not. What's the difference between a spore and a dead organism? A dead organism has water but no life. So is that related to complexity or scale?
Yeah, super nice question, right? So what is life? We come back to what is life. Um, Goes back to this definition, actually. It's a nice circular argument, and I think it's a very poor definition of life. But a spore, by all definitions of life, is not alive. It doesn't do any of these things, but it can come back to doing these things. So, in some ways, maybe the definition of life is having this ability at some point of time and regaining this ability to do all these things. And that's a spore. A spore is essentially completely suspended animation. It has no signs of life and no features of life, but it still has DNA. And it has intact DNA and the complete genome of that organism, which means if it comes back to life, it can replicate and divide and do all the things that you need to do to be alive. So, but once you're dead, first of all, your chromosomes or your DNA fragments, and it can't be repaired, and so you can't come back to life. So that's really that cusp of living and death. That's how I would think about it.
Right. So I guess it's something about the potential in some way.
You could imagine it to be that way.
Yeah. So another question is about hibernation.
Yeah.
So during hibernation, do proteins decrease their functionality or expression, or the expression is reduced to
Yeah.
induce a slow metabolic rate?
Yeah, so it's a good question. So hibernation is a slow conditioned process, right? You don't Animals don't just go to sleep; they prepare for hibernation. And what these lovely proteomic studies now, they've been really nicely done in the Colorado ground squirrel, which hibernates or goes into torpor hibernation. What they see is classes of proteins systematically reduce in expression. So as you lower temperature anyway, metabolism slows down, protein synthesis slows down. But there are a few proteins that go up in expression, and uh, these are mostly proteins related to removing oxidative stress and few chaperones. So you have classes of proteins that are made to protect the other proteins, but most proteins go down in their expression. And that is, of course, a function of the slowed metabolism that comes with decreasing temperature and sleep. Yeah. And there's, I think, another nice question: is sleep related to hibernation? Yeah, and the time scale is different. So this is great. This is a superb question because uh, this is exactly what people in chronobiology and people in circadian biology, so these are people who are very interested in aspects related to sleep, are rapidly realizing that a lot of features of sleep are just exaggerated and amplified as you get to hibernation, with this fact of managing the time scales, which means you need to have sleep doesn't need as many extensive regulated processes to turn up and turn down protein synthesis, turn down metabolism to this extent. So really, hibernation is a extreme extension of sleep with some differences. But what we are going to learn in the coming years is how sleep and hibernation are different, and how can we achieve controllable hibernation, just like we can now control sleep.
Yeah, that definitely sounds like something, interesting to study. If anyone here wants to go ahead and study that in the future and get back to us.
Yeah,
And
or if anybody wants to study it and can give us some money, we will study it in our lab, and you can come and join our lab.
sure, there's a plug for the lab.
Yeah.
So coming to some of the questions, someone wants to know, Devashri asks, how do sugars like trehalose behave in polar solvents other than water?
Um, it's an interesting question. Um, so they, I mean, with many polar solvents, you'll have many aspects of sugars behaving the same. But you have other aspects like pH and so on. So in slightly acidic environments, sugars will get hydrolyzed very quickly. So there is water, and any other polar solvent will also have some water in it. But also some of these features, like slowly forming these nice gels or semi-crystalline structures, all of that doesn't happen at the same rate and the same consistency as it does in water. So of course, this is again a circular argument, because life is all about water, and you have water and you have these sugars that help you deal with water. So I mean, it is a kind of a theoretical argument, and so nobody is bothered to hammer out the details of how exactly they behave in non-aqueous or non-polar, non-aqueous other polar solvents and so on. So I mean, yeah, debate.
Okay, cool. So there's another question about, I think, humans. For instance, as a follow-up to the question about what happens to our cells
Yeah.
when there's a lack of water, application molecules like trehalose protect our outermost cells from drying up and protect
Yeah,
the inner cells.
and
Something
make a
like,
super moisturizer.
yeah,
Well,
super effective moisturizer.
uh, so actually, in theory, you could use sucrose as a moisturizer. You just attract a lot of ants and so on, so it might not be as much fun. But In fact, you do know that in a lot of cosmetic treatments, you mix sugar with lemon juice and so on. A lot of that seems to improve skin tone. And there's a good reason for that. A lot of its properties and behaviors do help cells a lot. So in theory, and you use glycerol, which is in some ways, a complex sugar. So um, yes, in theory you could. It just won't be very pleasant because you'd be kind of a little too sweet tasting and attract a lot of discomfort due to other reasons, including allowing microbes to grow on your skin. But it works. It works. Yeah.
Okay. So someone else also is curious about humans. They ask, are there examples when a few cells in multicellular organisms undergo suspended animation and while others stay alive?
Yeah. It's actually a nice question. So, for example, in the worm that survives desiccation, do all the cells need to be protected or do only some? And can the worm revive after that? Um, there are a lot of cells that actually don't fully undergo desiccation and they do die. At least that's at a crude observational microscopic level, but the organism does survive. But there does seem to be a set of core cells that have to be fully protected and core organs that have to be fully protected. Now, how this kind of decision making happens in the organism is really not clear, and it's much more complex in plants where they have a bunch of cells that die, but the plant still revives. We have no idea how that happens, but there are absolutely examples in multicellular organisms of some cells entering suspended animations while many cells just kind of give up. So how that happens, no idea.
Okay. We have a few more questions. I think we can probably take one or two of them. Um, Maybe we can answer this one. So this is about what happens uh, when you thaw sperm after they've been frozen.
Yeah, so um, you know, sperm are interesting in that they are full of not trehalose, but they are full of proteins that would qualify as intrinsically disordered proteins. And they also have another huge amount of another set of molecules called polyamines, which also can behave in ways similar to some of these sugars and some of these proteins in that they can form meshes and gels. So it looks like sperm effectively is a rather well protected entity if you want to freeze it. Um, and it seems to be able to manage quite well. And so you can use it, for example, for freezing and use for artificial insemination without doing too much to protect it. You take some other cells and do treat it the same way, and they'd all die. So clearly, now this is interesting because most mammalian cells cannot easily survive extended freezing or drying down, but sperm can. So it's kind of unusual, and in that it has remnants of uh, in many ways, it's like a microbial cell. It's a cell on its own, and it does things that other cells don't. So in theory, yeah, it's interesting, right? Yeah,
Yeah, and there's a question about the formation of an endospore. So when it's formed, is all cellular material compressed into the endospore, or
yeah,
can the cell selectively discard things it doesn't need?
yeah. No, that's a nice question. Actually, it discards almost everything. It forms a compact little globule around the DNA. So all the DNA, the entire chromatin is condensed, and it has a globule around it with a very little bit of proteins around it. So presumably that set of proteins has enough of the transcription and translation machinery and some metabolic enzymes, and that's about it. But that's enough to get the spore started once nutrients come back, and then you start making more and more proteins and expanding them. So uh, about 90% of the cytoplasm is discarded when you have endospores. It's just condensed. What's left is condensed to a little ball that's mostly the genetic material.
Okay, cool. I guess we'll come to one last question because I think the video you shared with the worms has triggered this. So, are small RNAs also involved in slowing of metabolism, especially for worms?
Yeah, so we don't know right now, but I think there are people who are now looking to see if you can find regulatory small RNAs. I'm assuming what Shubham's asking is, are there regulatory small RNAs like we have microRNAs and so on that control this? And of course, worms have very nice RNA, regulatory RNA metabolism. So I think now people are trying to look and see if there are small RNAs in the Dower form of the worm which can survive all of these conditions. So we haven't found them yet, but the beauty of that kind of regulation is it's a nice way to get specific regulation in some contexts. So presumably the answer will be yes.
Okay, awesome. I think we are done with questions unless you want to return to any of those that were open earlier.
There's one question. There's some rumor that microbes have been found on meteorites, um, which give evidence for the extraterrestrial life. Well, that's the popular trope in all science fiction, and a lot of astrobiologists think that's plausible because essentially now spores are as close to indestructible as you can get. So, supposing there's a spore buried inside a meteorite, it might even survive uh, interstellar travel. I mean, if you can revive a spore from a fossilized bee from 30 million years ago, it is quite possible. So it is plausible theoretically. Now, of course, whether that happened or not, or whether there are microbes on Mars, we don't know. So there's no definitive evidence for that. But 20 years ago, if you'd said you can revive bacteria from Jurassic bee, you would laugh. So never say never.
Right. Okay. Thanks so much, Sunil. I think I'll hand it over to Mahin if she wants to sort of wrap up, or I guess we can end here. Thank you so much for
No,
answering.
hi. I'm so sorry. I was talking to myself because I was muted. No, I was saying, Sunil, I was just thinking to myself while you were speaking that it has been such a long time since I saw somebody so enjoy themselves when they spoke about the science that they did, and it was so delightful. And I really hope that everybody else who's on this call today also got a sense of that joy and that delight that you shared with us. So thank you so much for your time. It was really, really enlightening.
Very welcome. I hope everyone had a good time. Thank you so much.
Thank you all. Bye. Have a good evening.